All right, so let's talk about meiosis in more detail now. We have some idea of what it's for and what should be happening during it. Let's now go into actual details of what the chromosomes are doing in meiosis, the process of meiosis, which happens in ovaries and testes and produces sperm and egg cells. Okay, a couple things I want you to watch for in this. Watch for differences between this and mitosis. Watch for that. Watch for places where there are sources of variation, where genes are being shuffled. Watch for that. And watch for at what point the cells go from being diploid to being haploid. So watch for that. Okay. So I'm going to start with a cell. I'm just going to draw circles. I'm going to have different things happening. And I'm just going to go with, I'm not going to ask for a whole lot of input. I'm just going to make it so that you can just listen and watch, okay? So I'm going to make some, let's say, I'm going to make a whole bunch of red in here, and I'm going to make a whole bunch of blue. Okay, and right now, this cell is an interphase, right? So this is a cell in interphase. Meiosis hasn't begun yet, and it has chromatin in here, right? Chromatin. So it has DNA that is, let's say the red DNA is from mom and the blue DNA is from dad. So red and blue DNA, and right now it's completely uncoiled and it's all in there. So as I start meiosis, you're going to see those chromosomes coming into line and forming X's and going into different places and doing different things. Okay, so now let's do prophase. Prophase I. Meiosis has begun. So let's say that the nuclear membrane has disappeared, and now I'm going to draw two pairs of chromosomes to make it simple. I'm going to draw a big pair and a little pair. And again, remember I said that I had, I think I'll go with a thinner one here. I have mom and dad homologous chromosomes here, right? So there's that. There's that. Okay. So those homologous chromosomes are close together. They're really close together. And now a couple things are going to happen. Late in prophase, after the chromosomes have coiled up into their formation, so they have their sister chromatids attached to the centromeres, and they get really close together. Okay. So during prophase, something very important happens called synapsis. And with synapsis, homologous pairs get very close, like they are here, and they exchange bits of themselves. This is called crossing over, right? So I'm going to show you what the before or after of this is going to look like. Let's say that that is what's going to happen to these two mom and dad homologous pairs. And if that happens, so we're still in prophase I, okay? Here's that same cell again. Okay, we're still in prophase I, we're in late prophase I. And what happens is I'm going to redraw the chromosomes after the synapsis has happened. So basically what's happening is this. If I draw the chromosomes are doing over here, the big mom and dad chromosome, they are kind of like, this one is coming over here, right? And there is a break right there, so that after the crossing over has happened, you now have bits of the chromosome that have exchanged places. Oops, okay, yes. And I'll do it for the little guy too. There. Okay, and then go into red. So that the father's chromatid arm down there, the paternal chromosome, now has exchanged a little bit with it. And a lot more, it can be a lot more complicated than that, but I'm just trying to make it simple so that you can see. Okay. What the final mix is. See, those two bits got exchanged on both of them, right? Okay, so that's one thing that can happen during prophase. And there's one more thing that's going to happen. Right, so still in very late prophase here. The other thing that can happen is that the homologs can change places entirely. This is called independent assortment, meaning that maternal and paternal homologs switch places. Switch places. Oh my gosh, I'm sorry. It is so late in the day and I'm tired. Switch places across the, well, what you would call the metaphase plate. Even though we're not in metaphase yet. This has to happen before the spindle fibers get involved, or it can't happen, right? And we're still in we're in late prophase, even pro-metaphase here. Late prophase 1. There's a lot that happens in the very beginning here. So that now you've got the maybe the top switched places and the bottom didn't. Right? So you've got this guy, you've got this guy, and then you've got that guy is still over here. But this guy is now over here. Right, okay. So now you've got lots of genes that have shuffled their way around, right? Because remember, with independent assortment, there aren't just 2 pairs of chromosomes in your cells. There are 23 pairs of chromosomes. So all along that plate, this vertical line here, right? Before the spindle fibers get connected, all along there, you have 23 pairs of chromosomes that are lined up next to each other, which again is very different than it is in mitosis. And some of them switch places and some of them don't. So lots of gene shuffling has gone on before you really even get started with meiosis. Okay, so now we're going to go into metaphase 1. And after this, it gets a little bit faster. Those were 2 really important things to draw, and it's hard to draw them. So now you have these chromosomes, and we're in metaphase 1. And chromosomes are going to be lined up. Okay, and you've got, I'm just going to do it like this because otherwise it's going to take me forever to do this. Okay, and yes, you have this, and you have that, and you have this, and you have that, there. So that doesn't go away. All right, so now you have spindle fibers that are going to come in, because spindle fibers need to attach, and you still have the centrioles here. Spindle fibers attached to those centromeres. There's the spindle fibers. There's metaphase. Again, very different than mitosis, because with mitosis, those chromosomes line up one in singletons, right? But in meiosis, the homologs are next to each other, and they're going to be pulled apart. Now remember, back up here, we started with a diploid cell, right? This cell is diploid, right? Question is, I'm going to ask you this at the end when I'm all finished. When did it become haploid? All right, so Anaphase I. Anaphase I, we get a little bit of elongation here, there, and the spindle fibers are pulling those chromosomes in two directions, right? And look at what they look like. You should notice something here. Did these centromeres break? You should say, no, Mrs. Murdoch, they didn't. Good for you. The centromeres didn't break. The chromosomes and their sister chromatids are intact here. That's another big difference between mitosis and meiosis. Okay, so Anaphase pulls them apart, and then you're going to have Telophase I. Telophase I, we're just going to kind of Telophase I. Okay. So you have a little guy here, big guy here. Big one, little one, little in there, little in there. Because the results of that original synapsis are still there. There we go. Okay. Next phase. Oh, come on. Of course, I'm going to have to go up here and do this now. Okay, I'm on meiosis II now. So now we've gone through telophase I. Now prophase II, we're going to kind of skip. Prophase II, nothing really happens here. You still have the same things going on. I'm just going to kind of draw the same cell. I can't remember if I kept everything. Okay. I'm just keeping the same diversity. All right. So prophase is pretty much the same. I'm going to go right into metaphase II. Nothing really happens in prophase II. Okay, so this is in metaphase II. You have a new set of spindle fibers that are going to attach themselves this time to the centromere of each chromosome. There, there. Metaphase II looks a lot like mitosis, ordinary metaphase. There, okay. And then the next thing that happens is Anaphase II. So Anaphase II, we're going to have the cell elongate in both cases. So here's Anaphase II. Second thing, okay. So in both cases we have movement of, this time we have movement of sister chromatids. this time, watch me to make sure I do this right. So here's a long red one. And here's the short red one. And here's another short red one. And here is a short red one. Okay. Here's a little blue one. And a little blue one. And this one we're going to say has the edge there. Here's a long blue one. Here's another long blue one. Just trying to make sure I keep it all right. So this one has the little thing and that one doesn't. Yeah. Okay. A bit more of this. That has a red edge to it. That one doesn't. And this one has a little red edge. Okay, there we go. Okay. There's Anaphase II. And then finally, Telophase II, the final cells. Right? You're going to make 4 cells in Telophase II. Right. That are going to have the following chromosomes. A long red homologue here. Oops, long red homologue here. Another one here. Short red homologue here. Another short one there. Okay. A little blue one here. Blue edge there. A little blue one there. Big blue one there. Blue edge there. Big blue one there. Okay. And that has a red bit. That has a red bit. There, that should be it. Okay. Okay. All right. So these final cells here at the bottom in Telophase II, diploid or haploid? If you said haploid, you are correct. Yes, they are haploid. Not only are they haploid, because none of these chromosomes have their homologous partner with them, but they have been scrambled a bit, right? This in particular, this cell here and this cell here have some recombinant chromosomes in them from the original crossing over that happened in prophase, and they still have that. They carried it all the way down. Okay, so here's the question though. If I go all the way back up through, where was the point when the cells became haploid? Did that happen here at Anaphase II? Or did it happen here at Anaphase I? It had to be one of them, right? It has to either be Anaphase I or Anaphase II where that happened. When we took them apart when they were the X's, did that separate the homologs, or did the final Anaphase separate them? And I can hear your opinions popping in. Let's see what you say. Okay, so what do you think? How many of you think Anaphase II, and how many of you think Anaphase I? This isn't necessarily easy. Okay, so the answer is it's Anaphase I. The division that comes first is actually the point where you're separating the mommy and daddy homologs from each other. You see? That's where you're separating the red from the blue. You see that? That after that point, all you really have are the sister chromatids. You have extra copies to put in the final cells, but you have separated your big B's from your little b's if they were there, right? You've separated your alleles that were here from the other alleles that were here. So Anaphase I, it's Anaphase I that is the place where you first have haploid cells, right? It was diploid here. It was still diploid. The red X and the blue X's were still together. As soon as you separate the paternal from the maternal X's, it's now haploid. And then after that, all you're really doing is separating the copies, the sister chromatids, right? So this up here, I'm sorry, this one kind of went backwards. This final division here this is still haploid, right? but this has the copies of the chromatids in it. Then you're still, at the end, you have, like, for example, if you had a big B on the big ones and little b's on the other ones, you still have those, right? But the only difference is you only have one copy of each. Okay, so that is meiosis, and those are my colored drawings of meiosis. Okay, so starting at the top, I'll just go back through it again. Meiosis, differences between meiosis and mitosis. It's the way the chromosomes line up along the first metaphase. It's the biggest difference. That and the sources of variation. And the fact that it goes from diploid to haploid. In mitosis, none of those things happen. So remember that we have interphase, and then they coil up, and you have, in prophase I, you have a couple big events that happen. And these are the sources of variation. In synapses and crossing over, you are exchanging bits of the chromosomes, and that scrambles up the genes. That's one source of variation. That's called crossing over. And the other source of variation is that the maternal and paternal chromosomes might completely switch places before phase I begins. And that makes for entire chromosomes being shuffled back and forth. Okay, so those are the two main sources of variation, and then metaphase I sets them up so that the chromosomes are going to be hooked to spindle fibers, and the homologs will be taken apart in Anaphase I and separated from each other, which achieves the reduction division that makes the resulting cells haploid from that point on. And then in the second meiosis steps, now you have prophase I, which really is neither here nor there. There's not much happening there. But then metaphase II is now going to pull those final sister chromatids apart. Anaphase II does that. And then the final cells are haploid with only one of each of the homologous pairs in them. And a lot of gene scrambling. Okay, so now finally, in a male, in the testes, what are all of these cells going to become? In a male human, the fate of all four, right? Yes. Fate of all four is to become sperm cells. Right? Okay. In a female, it's a bit different. In a female, one becomes an egg or ovum, and three become something called polar bodies, whose only purpose is to get rid of the extra chromosomes so that this one will go on to possibly become fertilized and become a zygote. Polar bodies disintegrate and are discarded, not needed. Okay. All right. So that is as much as I'm going to share with that.